is coming up in the audience Q&A. Doug, you just mentioned some specifically, regulatory hurdles and bottlenecks. And I think that this question from the audience, actually, we can address some of that. How can we evolve our translational approaches in the cell and gene therapy space to more confidently enter the clinic and not simply rush into phase ones by performing simple minimum criterion analyses mandated by the FDA currently? And this person says, which I think we can agree, are still falling behind. So, Doug, I'll let you take that one first. And then, Greg, I'd love for you to weigh in on this too.
Yeah, I think one of the huge challenges here is something that I mentioned earlier, which is the lack of really strong animal models for a lot of these diseases. If you don't have a really strong phenotype in your animal model, you can still get really good biomarker data. Often the biochemical processes are usually the same, but if you're trying to translate from preclinical to clinical in terms of behavioral changes or clinical responses, it can be very difficult to sort of build that model in your preclinical experiments because the physical changes in the animal just aren't there. So I'll give an example. One of the diseases that has been targeted by ex vivo lentiviral gene therapy is metachromatic leukodystrophy. The animal model for that is an arsen knockout mouse that is completely null for expression of the aryl sulfatase A protein, no enzymatic activity whatsoever. In human patients, that disease shows up somewhere before 2 years of age, and many of those kids don't make it out of childhood. So it's a quick onset, very severe disease. In the mice, you don't really even see any physical measurable changes in those mice until they're about a year old, and most of them will live, just as long as you expect a wild type mouse to live. You have to have very large studies, very highly powered, in order to be able to actually see the physical changes that are happening in those mice. Whereas the biochemical stuff is all very cut and dry and clear very early on. So in that type of situation, your phase 1 study is really the first time you get a peek into how those biochemical biomarkers actually translate into an efficacy in the patients. And that's a huge challenge. I think, one of the things that I've seen recently is that industry and KOLs, especially these centers of excellence for specific diseases, are investing early on in natural history studies, which is hugely helpful because having that baseline data in patients really allows you to make a lot more of the first couple of patients that you treat in your phase 1 study. So in a case like metachromatic leukodystrophy, having a body of natural history data that's accumulated within the context of a clinical trial, as opposed to gleaned out of visits that patients normally make to their doctors. So data that's collected in a structured way with measures of biomarkers that are going to be relevant to later clinical trials that include a treatment factor can be hugely helpful. And I'm seeing more and more that industry and KOLs and physicians are collaborating to make those sorts of studies happen, and then making that data available so that it can be used to support trials that actually have a therapeutic component.
Good. Okay. Thank you. And Greg, I want to give you the opportunity to weigh in on this same question, which is, like we said, evolving our translational approaches in the cell and gene therapy space more confidently into the clinic. What do you think?
Yeah, thanks, Erin. If I understand the questioner right, they're suggesting that the minimum bar before entering the clinic set by the FDA is too low, and that the questioner's own opinion, the bar should be higher. But obviously, when you work for biotech or pharma, you can set the bar as high as you want over the FDA's bar. So I think it really comes back on the questioner and a leadership opportunity to step up to senior management, to step up to the development team and say, I don't think we should go forward into the clinic with the minimum requirements. I think we need to raise the bar even higher so we can more confidently enter the clinic. And then I would put it back on the questioner to develop their business development skills. Because so many folks like myself in the translational space are very left-brained, very analytical, very, data-driven. But oftentimes what senior management needs, what your assay development team needs, is somebody who can explain to them why the FDA thresholds are too low, why the organization is better than that, and why we should do the additional assays, how they may de-risk the assets, how they may de-risk the phase 1 clinical study, and the return on investment. Now, obviously, that's not always the easiest job for a translational scientist like myself, like the questioner. So maybe that's an opportunity to partner with somebody else in the organization who has those skills in abundance and who's willing to, form up with you. It adds cost, it adds time, it adds complexity to your, IND enabling tox paradigm. But it clearly sounds from the questioner that they feel deep down inside that it's worth it. And there's nothing more valuable than a passionate scientist who believes in setting the bar very high for themselves and their organization.
Oh, great advice. Thanks, Greg. That's wonderful. One more question here I want to make sure we get to. Given the cell and gene therapy analytical development endpoint assays and translational clinical assays are often looking at the same things, how do you maneuver your company's infrastructure to have the AD and biomarker translational groups communicate and work together better? Greg, you're perfect
Sure.
for this one. Go right ahead.
Well, again, this is one of the systemic and endemic challenges to working in a big pharma environment. And I can't tell you enough how much I endorse the book by Safi Bahcall, ex-Princeton, ex-McKinsey. It came out 3 or 4 years ago. And whoever wrote this question, I hope you have a copy of Safi's book called Loon Shots sitting on your bedside nightstand. It directly addresses the challenges of drug development in a large pharma environment when every department head holds the veto. And it only takes one department head, one functional area head to say, I'm not sure. Everything stops. So in this current example here, you're talking about an assay that works both for, say, product release testing, and then the translational group would use the same assay in vivo in patients and subjects to measure drug product, if you will. So I can't give you a great answer how to do that in big pharma because let's be honest, that's why big pharma struggles in terms of productivity. That's why it takes big pharma, what is the latest number, $4 billion to develop a drug, whereas biotechs can do it for $1 billion. And this exactly gets to the heart of that point. So I can just say that in the biotech sphere in general, what we generally do is we take that analytical release assay once it's been validated and qualified, GLP, GMP, GCP, et cetera, and we generally put it at a third-party CRO, somebody that's trained, qualified, and tested. But now it's neither owned by the manufacturing group or the translational group, but both groups can avail themselves of those testing facilities and resources. And at the end of the day, you've now reduced that to practice. It's now a fee-for-service assay. They turn the crank, they generate high-quality data, and both parts of the organization get the benefit from it.
Good. Okay. Could you repeat the name of that book too, please? Make sure we heard it correctly.
Sure. It's Loon Shots. It's kind of a take on the old phrase moonshot, but
Yep,
loon
sure.
as in the bird.
Got it. Okay, thanks. And Doug, I want to make sure you also have the opportunity to weigh in to give your response to this particular audience member's question.
Yeah, well, I've now had the privilege of working at 2 biotech companies where the solution to this particular problem was to have a centralized analytical development group. So at Affinia Therapeutics, I ran that group, and at AVROBIO, I worked very closely with that group. But the idea here is that, traditionally, analytical development has been under the manufacturing umbrella, and translational medicine has had scientists that do assay development for preclinical and translational and clinical assays, and those have been very separated. But at both Affinia and AVROBIO, those groups do both. So the analytical development group is responsible for developing the assays that are being used in manufacturing to characterize drug product, but they're also responsible for developing assays that are characterizing the performance of the drug in the animal models and doing early development, at least on assays that are going into the clinic that may eventually get transferred out to a CRO. And I think that's a good solution because it provides for a pool of people that have a very good understanding of all of the assays that are being used to support the development of the drug. You have internal experts that can troubleshoot, different aspects of those assays, and it also sort of forces at least one group of people to make the connection between the manufacturing assays and the preclinical and translational assays. And having that connection really helps to bridge the gap and make sure that those groups are communicating appropriately. So currently at AVROBIO, the centralized analytical development group, the translational medicine group, the preclinical group, and the manufacturing analytics group all meet regularly. And those meetings are specifically dedicated to talking about analytical methods and developing strategies to make sure that we're aligned across the entire organization on the development of those, and for assays in particular that are used across all three functions, aligning as closely as possible on platforms and validation criteria and performance criteria, and then making sure that when assays are transferred out, that they're, as Greg said, sort of available for all the groups that might need them, and properly validated for all the different matrices that might be coming to the CRO performing that assay.